To officially commence our conference and to introduce our esteemed chief guest, it is my pleasure to invite the chair of the conference, Professor Geethanjali Satyadas, who has been an integral part of the planning and execution of this event, to the stage, please.
Good morning, ladies and gentlemen. I am both privileged and honored to introduce Professor Thomas Coffman, a globally acclaimed expert, to this audience. Professor Thomas Coffman is the Dean of the Duke NUS Medical School in Singapore and the James R. Clapp Professor of Medicine at Duke University School of Medicine. Professor Coffman graduated from the University of Pennsylvania and obtained his MD from the Ohio State University School of Medicine. Before moving to Singapore, Professor Coffman held several leadership positions at Duke, including the Chief of Division of Nephrology, Vice Chair of the Department of Medicine, and Founding Director of the Duke Cardiovascular Research Center. He was also the Founding Director of the Duke NUS Cardiovascular and Metabolic Disorders Signature Research Program. Professor Coffman serves on the boards of directors for Singapore Health Services, the largest public health hospital cluster in Singapore, the Singapore Eye Research Institute, the Singapore National Medical Research Council, and the King Faisal Specialty Hospital and Research Center in Saudi Arabia. He also serves as the Medical Advisory Committee Chairman at the National Kidney Foundation, Singapore. An international leader in the field of nephrology, Professor Coffman was a counselor and past president of the American Society of Nephrology and served as the treasurer for the International Society of Nephrology. He is a member of the American Society for Clinical Investigation and Association of American Physicians and served as a member of the Nephrology Subspecialty Board of the American Board of Internal Medicine. He has held a number of editorial positions, including Associate Editor for the Journal of Clinical Investigation, and currently serves on the Editorial Advisory Board of Cell Metabolism. Professor Coffman has been a productive researcher, publishing over 200 scientific papers and H-index of 84, and he has been funded for many years by the US National Institute of Health. His laboratory has made seminal contribution to understanding hypertension, the renin-angiotensin system, and the diabetic nephropathy. In 2014, he received the Excellence Award for Hypertension Research from the American Heart Association for making a major impact in the field of hypertension, and whose research has contributed to the improved treatment and greater understanding of high blood pressure. He has also received several other awards in the field of nephrology and cardiology. Professor Coffman currently serves as the lead investigator for DYNAMO, Diabetic Study of Nephropathy and Other Microvascular Complications, a $25 million large collaborative grant funded by the National Medical Research Council of Singapore. Ladies and gentlemen, let us all welcome Professor Thomas Coffman to deliver the keynote address of the 2024 Rcfmj conference on using omics technology to address the problem of diabetic nephropathy. Over to you.
Thanks very much for the invitation to be here. Really delighted to be here for three things. One, to be a part of this high quality and exciting research symposium. Was glad to be here for the opening of the new clinical trials unit yesterday. And also to be here on behalf of Duke NUS Medical School and the Sing Health Duke NUS Global Health Institute to reaffirm and celebrate our partnership with the University of Jaffna as one of our regional collaborating centers as part of a global health network. And we spent yesterday afternoon reviewing the range of really exciting, impactful projects that are going on as a part of this collaboration, which I think hopefully this is the beginning of a beautiful friendship that's going to last for many years, and I think will enrich both partners, which is that's the kind of partnership you want, that where both sides will benefit. And I think this is one that we're completely committed to and really look forward to seeing it flourish in the future. Now, I understand there are some medical students here today. Is that true? Can the medical students raise their hands so we can see who you are? Yeah, wow, quite a good group. First of all, I want you to remember the lecture that you just heard from Dr. Sriran, which is basically a beautiful guide for how to conduct yourselves as you pursue your career in medicine. Really I've never heard it laid out more beautifully, so fantastic. And very fortunate for all of you all to be here. Now, I'm going to try in my own way to talk a little bit about one facet of that, and that is how research might contribute and can contribute to the improvement of medicine, and also can be incorporated as part of a career of a doctor in a way that's fulfilling and useful, but also gives back to society. So, get up my phone just so I don't go on too long. But so I have two parts to my talk, basically. I'm going to talk a little, just in the beginning, a little bit about Duke NUS Medical School, where I come from and our team comes from, just to give you a little bit of perspective of who we are and what we're doing. What we bring to the partnership here, and how we support impactful medical research as part of our mission. And then I'm going to talk a little bit about a project that I'm involved in. Yes, as you heard, in full disclosure, I'm a nephrologist, and thanks very much for that generous introduction, by the way. But we focused on kidney disease, which is a big public health problem in Singapore. So, just a little bit about Duke NUS. So Duke NUS is a partnership between two great world universities, Duke University in the United States, where I spent many years before coming to Singapore, and then the National University of Singapore. Like this is your 50th anniversary, this year is actually our 20th anniversary. So we're younger, we're going forward, trying hard, but it is actually a momentous year for us. We're a public school within Singapore, supported by Singapore's Ministry of Education and Ministry of Health, but a partnership, academic partnership between Duke and NUS. We were brought to Singapore specifically to begin a graduate entry, American-style medical school. So our students have to have a university degree to be able to be admitted. Many come with advanced degrees, but they're older and bring different kinds of life experiences to the practice of medicine. And specifically, as a research-intensive medical school, one of our objectives was actually to educate medical students, some of whom would go on and pursue careers as clinician researchers. So clinicians who understand the practice of medicine, but also do research, and therefore can bring research principles into their, and research advances more readily into their clinical practice. Our students come from universities all over the world. This is just a list of some of those. Our two local universities, the biggest suppliers, but really all over the schools all over, including from India. We have, this is some of our just sort of facts and figures. As I said, we were started in 2005, so we're 20 years old. Actually, by coincidence, my own birthday was yesterday, and I want to appreciate, I'm not going to put the number up here, but I appreciate the very nice celebration and the unbelievable cake that was created. And too bad we didn't bring it today. There's probably enough cake to feed everybody in the auditorium. But so 20 years old, as I said, our students come with university degrees. Many come with advanced degrees, sometimes masters and PhDs. We have a research component integrated into our medical school curriculum. So our students published a lot of papers. You can see over the years, over 700 publications in total. We do have a strong research base in the school, which really, again, we're trying to move medicine forward, but also provides an atmosphere where students are educated, are exposed to the principles and the activity and the excitement of research. We don't have the usual basic science departments because we're a new school. We don't have the usual basic science departments, so no Department of Anatomy, Biochemistry, Physiology. Instead, we have our discovery research is organized in 5 signature research programs that are really disease-focused and focused on diseases and health problems that are important in the public health of Singapore. And they're listed here. Emerging infectious diseases, which was obviously relevant during COVID, cardiovascular metabolic disorders, which the work I'm going to talk about is really based in that program, which is where obesity, diabetes, and cardiovascular risk sort of come together. Neuroscience and behavioral disorders, focusing on the neurologic disorders of aging. Singapore has a very rapidly aging population. Cancer and stem cell biology, we focus on cancers in the region, particularly things like hepatic cancer, hepatobiliary cancers, viral-associated cancers. And then a health services and systems research program, which is really kind of like a mini school of public health, where we go more ground level to implement new practices and influence the way medicine is practiced on the ground. We publish a lot of papers. We have a lot of grants and collaborations. But as Dr. Sriran just mentioned, doing research in medicine is not about publishing papers or getting grants. It's around really having impact. And one of the ways that we've tried to facilitate our research having impact is to develop and enhance the commercialization of our research to help move discoveries from the laboratory out into the public where they can really help patients. And to do that, you often have to have commercial partners, and there's a whole process involved to do that is not typically part of medical school curriculum. So something we've implemented, and it's resulted in a number of licensing deals and startups and to develop new drugs and diagnostic procedures that we hope will, in the future, impact our patients. Very importantly, as part of the school, we have a strong academic medicine partnership with Sing Health, and I'm here with many Sing Health colleagues today as a manifestation of that. And the whole idea here is that, we need a place for our students to train, to do their clinical training. And the majority of that happens within the Sing Health institutions, which are pictured here. And we're just across the road from each other, so it's quite convenient. Unlike you all, I think a little bit further away from the hospitals. But most of our students get their clinical training and have the benefits of being in this outstanding, high quality, high volume clinical care system. But the other part of this, from the standpoint of academic medicine, it allows us a ready partner to be able to move questions that are generated or that are solved in the lab and new discoveries, then can bring them across into the clinical system to the bedside to help patients. And then vice versa. The questions that arise at the bedside then can be brought back to the research lab for analysis and hopefully to try and solve clinical problems and develop new therapies. Now I'm going to talk to you about one. I'm going to talk now, sort of move into the research part of my talk and talk about one problem that we've been interested in. And keep myself dressed. This is a complicated outfit, I got to say. But I'm going to talk about a big problem around the world and particularly in Asia, and that's diabetes and diabetic complications. And I'm a nephrologist and particularly interested in diabetic kidney disease, which is an important complication of diabetes. And you can see the numbers here is that they're a substantial number of the global population of diabetes is in this region, in Asia, Southeast and South Asia. Just to remind you that there are many complications, a number of important complications of diabetes. Kidney disease is one of the most devastating, and it's classified, diabetes in terms of complications is a disease of blood vessels, and there are microvascular complications in the small blood vessels and macrovascular complications in larger blood vessels. So kidney disease and also eye disease are both manifestations of the small blood vessel disease of diabetes, and they go together actually. They're quite and often quite linked clinically. And then also diabetes causes coronary disease and other large vessel disease, which are separate processes but also related. I'm really going to focus today mostly on kidney disease, also a little bit on eye disease. So it turns out that diabetic kidney disease is a very common problem in Singapore. So you probably can't see the letters here, but this is from the United States Renal Data Service, which collects information on kidney disease around the world, looking at the incidence of diabetes as a cause of end-stage kidney failure. This is in 2021. Because you can't see the writing here, this box here is the incidence of diabetes as a cause of end-stage kidney failure in Singapore. And number 3, really near the top of the list here when you look globally. It's also, if you just look across the board at the adjusted prevalence per population of end-stage kidney failure, Singapore is number 5 in the world. Most of this is driven by diabetic kidney disease, which is exceedingly common. And in fact, in 2021, more than 2/3 of the cases of end-stage kidney failure in Singapore were caused by diabetes. It's a huge medical problem, significant unmet need in terms of treatment. And as you may know, treatment of people with end-stage kidney failures with dialysis, very expensive and tremendously impactful in a negative way on families and patients that are involved. Within Singapore, there are 3 main ethnic groups. And turns out there's a differential risk for the development of kidney disease with diabetes between these 3 groups, with Malays being the highest, Chinese, intermediate, and Asian Indians, in fact, have the lower incidence of kidney or risk for kidney disease, higher risk for heart disease. But this differential risk between the groups suggests that there may be genetic factors as one contributor to development of risk for kidney disease and diabetes. And that's one of the things that we're interested in the program I'm going to tell you about. So that program is called Dynamo, Diabetes Study in Nephropathy and Other Microvascular Complications. We cheated on the acronym, but Dynamo we thought sounded really cool and forceful. And we're actually in the second phase of this. It turns out, even though people have been studying diabetic kidney disease for many years, for decades, but really haven't been able to get to the crux of what the main molecular causes of kidney disease and diabetes are. And so the approach that we took was to say, come on, let's step back and try and utilize some modern, powerful technologies to see if we can address this. The omics technologies, where we measure large numbers of metabolites, genomics and genetics, where we look at genetic sequences, gene expression. So we can approach these. We don't have to come up with a smart hypothesis for this. We use these technologies, which are pretty much agnostic, and we look at the patterns, whether it's metabolites or DNA sequences, and look and see if they can suggest novel factors that may be causing susceptibility or resistance to kidney disease, and see if we can exploit those for understanding pathophysiology, but then also to use those for perhaps diagnosis or as targets for treatment. So that's been the basic gist of the program. And as I said, to try and address this unmet need by identifying new mechanistic pathways, discovering new targets for treatment, and new biomarkers and strategy for allowing us to identify patients at risk. Because as you may know, of all the people who get diabetes and have it for many years, only about 30 or 40% will go on and develop kidney disease. And it's very hard to find those patients prospectively until they actually begin to manifest signs of kidney disease. So if we had markers that we could use to identify that group at risk, we could stratify them for more intensive therapies and more specific therapies to prevent the development of over kidney disease. So while the program is really focused in Singapore and involves all of the medical schools and hospital clusters in Singapore, it's actually an international collaboration across 6 countries. We have many big patient cohorts, well, I'll tell you a little bit about in a minute, many institutions and investigators that are, and it's a multidisciplinary group. So there are people doing basic science, they're clinicians, both endocrinologists, nephrologists, cardiologists, data scientists. We have some engineers involved. So we bring together, perspectives from many different disciplines to focus on this problem to see if we can come up with new solutions and insights. what we did in the first, so I said we're in the second iteration, the second 5 years of this program. What we did in the first iteration was to put together large numbers of patients, diabetics, who were enriched for diabetes complications, in particular kidney disease. So you can see on the top here, we have a number of subjects that are part of this, brought in by various investigators that have had blood and urine samples and other kind of biologic specimens. And then we took those specimens and just applied the various omics approaches that we had available. So we've done whole genome sequencing there, so sequenced the whole genome and particularly focused on individuals of Chinese descent and Malay descent, because the ones with the highest risk. And we have a group of diabetics, half of whom get kidney disease, half who don't. And then we're comparing the various genetic variants on both sides and look for variants that are enriched for either development of kidney disease or resistance. And then the fact that we have two different ancestries allows us to make more powerful inferences about whether these variations in DNA that we see have any specific cause. GWAS arrays to be able to do genome-wide association studies. Within the Singapore Public Hospital System, and the Singapore National Eye Center has for years been taking retinal photographs. And so we've used these in collaboration with the scientists from CIRI to use artificial intelligence to analyze the retinal images and then to try and identify patterns that are associated with specific risk for kidney disease. I'll show you a little bit of data about that. And then metabolomics, which is where we use basically GCMS platforms where we can measure very large numbers of metabolites in small biological samples. In this case, I'm going to tell you about, or just mention some studies we've done using a targeted metabolomics platform that measures known metabolites across the various energy generating pathways. And diabetes, in fact, is a metabolic disease. So we think that changes in metabolism might be particularly useful in trying to identify and understand kidney disease. Also, to use some proteomics, and we're just beginning to do kidney biopsies from individuals who have kidney disease and apply these techniques to the biopsy specimens. Won't go through all this, but these are the 5 theme leaders within the LCG from Lim Soo Chi, who's from NHG, Kutek Bot Hospital, Gavin Tan for theme 3, which looks at the association between kidney and the eye as an ophthalmologist at the eye center. Tai Shang, who's an endocrinologist and geneticist, looking at the genetics of common variants and their contribution to kidney disease. And then Enrico Petretto for theme 5, who's a brilliant data analytics person who's looking for rare genetic variants and their contributions. So just a few examples of the kind of information that's emerged from this. And this first study I'm going to tell you about actually came from our group, where this is one of those studies where we looked at just the patterns of metabolites in comparing individual or either animal models or patients with diabetes with and without kidney disease. And what we found in both animals and in patients with diabetes who go on to develop kidney disease is this very characteristic, surprising metabolic change, which involved the TCA cycle. Now, I was, to the medical students here, I shouldn't say this, but I really didn't like biochemistry when I was a medical student, and so having to go back and revisit all these pathways, it was a little bit difficult, but now I see why it's important. And in fact, we found in this important energy generating pathway, we found a signature of changes in metabolites that identified people that were going to go ahead and develop kidney disease. And the most prominent one was an increase in lactate. And this is the lactate, we saw it in the kidney, but we also see it in urine as a reflection of kidney metabolism. Now, lactate, we think about, if we climb too many stairs and our muscles start to burn, that's lactate. But it turns out that it seems to be very important in the development of kidney disease and actually tracks with this broader metabolic abnormality in the TCA cycle that's associated with kidney disease development. Most importantly, what we found here when we looked at a bigger group of individuals with diabetes who went on to develop kidney disease, that a high lactate level was associated with progression of kidney disease and development of end-stage kidney failure, which is very surprising, but statistically highly significant, and it suggests, one, that measuring lactate might be something that we could do to identify people who are going to go on to have risk for kidney disease. But two, if we could figure out why lactate is made, these abnormal lactate metabolism happens, maybe we could do something to address the metabolic defect and help protect the kidney. And then finally, it turns out that in cancer, lactate itself has detrimental effects through binding to a specific receptor. So it may be that the generation of lactate itself may be contributing to kidney injury. So going forward, we're exploring all these possibilities sort of in this next phase. But I hope an example of how this kind of just broadly, without any hypothesis, broad measurements of metabolites can actually provide significant and interesting biological insights. Something maybe a little more clinically approachable, which has been done by the Theme 2 investigators under the guidance of Lim Soo Chi, who's an accomplished endocrinologist. They used clinical factors to try and segment and identify subpopulations of people with diabetes that might be able to be transmitted into or translated into changes in risk. And they used a range of different clinical markers, but also genetics and lipidomics, lipidomics is like metabolomics. Instead of measuring general metabolites, you're actually measuring lipid compounds. Not a very imaginative use of language here, but what they found is by combining these all together, they found three groups that were very similar and actually comprised most of the diabetics that they saw in their clinics, but with different sort of pathophysiology. So MOD was the mildly obese diabetics. What they call MARD2 was people who had mild age-related diabetics, but with insulin deficiency. And then the third category was severe insulin-resistant diabetes and relatively insulin deficient. And by categorizing these, again, most people fell into one of these three categories. And then when they looked at risk of either heart disease or kidney disease, that there was a differential risk. And those who had the severe insulin-resistant diabetes had a much higher risk of developing heart and kidney disease, whereas the people with mild obese diabetics had less of a risk. And so now they're doing this proactively in their clinics. They're doing this segmentation based on clinical variables and then customizing therapeutic regimens based on what they've identified as the pathophysiology equivalent to try and see whether they can improve outcomes. So another way of combining clinical variables with these new technologies to be able to stratify risk and change the way we treat people. Getting close to the end, so I'm just going to, this is the eye, kidney part of this is from Siri, and in a nutshell, what they've been able to do is to use AI to analyze retinal photographs, large numbers of people with diabetes, and they found specific AI algorithms that can identify individuals who are at risk for kidney disease. And you know, one of the things with AI is that AI does what AI does. We don't always understand how these separations are made, but it does seem to be, be able to do this in a way that's statistically significant and more powerful. And so we're trying to understand the basis of these, and then to test prospectively whether we can use these kind of AI algorithms to improve our diagnosis. And then finally, I'll just give an overview of we're using kind of a multi-pronged approach for trying to understand the genetics. And as I told you in the beginning, that there does seem to be this strong difference in risk between the various ethnic groups within Singapore. And so we're taking two different approaches. One, as I said, to do whole genome sequencing, which looks at every every part of the DNA sequence and look for variants that are associated with people who develop kidney disease who don't. And then we're doing GWAS, or genome-wide association studies, looking for common variants in DNA and how they segregate with kidney disease without. We have some animal models that we're looking at. And then the question is, with these genetic studies, is that you find a variant, you find a difference in DNA that identifies or is associated with the disease. Doesn't mean anything. Out of these billions of bases, how do you figure out which of these variants are really disease-causing? And we have some interesting ways of screening those. One is in flies, in Drosophila, where you can very easily create genetic variants and then look at their effects on whatever system you're looking at. Turns out flies don't have kidneys, but they do have these cells called nephrocytes, which are analogous to the cells that line the filter of human kidneys. And so we're using those to look for variants that affect structure and function in those cells. And then we also have a collaborator at NTU that makes kidney organoids from stem cells, from skin cells, from individuals. And again, we can look at the effects of variants in kidney structures within the organoid, again, to look to see whether or not these turn out to be functional. I'm going to skip over this part, and just to conclude, so just because I love the structure of the kidney, I'm going to torture you by showing you this picture, but this is actually a picture of a glomerulus in an individual with diabetic kidney disease, where you see a big nodule, the Kimmel-Still-Wilson nodule, which is sort of pathognomonic, but not necessary to make the diagnosis for kidney disease, just for the medical students, a little pearl for you there. But otherwise, you see this nodular accumulation of matrix material, which eventually takes over and stops the filter and causes end-stage kidney failure. And so really our target is to try and prevent these sort of lesions from forming, or for individuals to have them regress and stop their progression. And it had, this disease, I told you what a difficult problem it is in Singapore, and it's actually become the most common cause of kidney failure across most of the developed world. So here's just to give the kudos to the bigger group of individuals who are involved in Dynamo for their outstanding work. And thank you very much. I'm very honored to be here today, very honored to be a part of this collaboration with the University of Jaffna, and thanks for your attention.